Hola, Team Bio. Okay, so today's screencast is going to be about genetic drift. So we've talked about some of the requirements for evolution. Remember, there must be variation within a population. That variation must be heritable, and then there must be differences in fitness among individuals in population due to that heritable variation. And then there must be some sort of competition and unequal survival and reproduction rates. And then, given all those conditions, we see changing allele frequencies in populations. And this is over time, and this is, in a sense, evolution. But a key assumption that we've made in this model system is that we are always selecting for the best alleles suited to the environment, or the alleles that are best suited to the environment. So essentially, that evolution is always leading to the most fit organisms surviving and reproducing. And that is actually not always true. There is this effect called genetic drift, which shows us that sometimes individuals, just by chance, leave behind a few more descendants and therefore a few more genes than other individuals. And so the genes of this next generation will happen to be the genes of the lucky individuals, but these individuals, the lucky ones that were left more descendants, are not necessarily the healthier or more fit individuals in the absence of whatever this disturbance was. So that is genetic drift. It happens to all populations, and basically there's no avoiding this kind of chance element to evolution. And I think I'll give you an example that will make this a little bit more clear, because that was kind of vague and not very well explained. So imagine, I'm going to add a photo here. Okay, imagine you have a population of beetles, and there is there's some with the green phenotype and some with the tan phenotype, and they're all within a certain area. And then in time 2, along comes the foot and just steps on 2 of the 3 green beetles that exist in this population. Now, this wasn't a person who was seeking out to destroy green beetles. It was just a person walking along, and unlucky chance for the green beetles, 2 of them were smushed. So then we move to this last panel here, and you see that there's only 1 more green beetle left, and 6 tan beetles. So we've changed the allele frequency within this population, but it has nothing to do with whether or not this green beetle was more fit than the tan beetle. Instead, it was just a random chance occurrence. You can imagine the same thing happening like in a forest. If half of the forest burns down, then the organisms that were in that half of the forest, those trees that were in that half of the forest, are gone. And they weren't any more fit than the trees on the other side of the forest. They just were in the wrong place at the wrong time, or a tsunami, or a tornado. All of these chance occurrences wipe out certain alleles, wipe out certain individuals, and then therefore change allele frequencies within the population. So, now as you can imagine, genetic drift has a stronger effect on steering evolution if the population is smaller. And the reason for that is there's a higher probability that, I guess I'll describe it this way. Okay, so imagine you have a coin. Here's a coin, and here's, no. Okay, this side is heads. There's a head on it. Okay, so imagine you have this coin, and I asked you to flip it 10 times. I don't think you would be too surprised if you got 3, if you had to flip it 10 times, you had 3 heads and 7 tails. But then I told you to flip it 100 times. I think you would be a little surprised if you started to see 30 heads and 70 tails. And if I asked you to flip it 1,000 times, you would be very surprised if you only had 300 heads and 700 tails, and so on and so forth. So, you get the idea here that when you have a small population, it's not unordinary to see a much wider distribution in terms of the number of heads and tails you see. However, as your population gets bigger, or number of flips gets more and more and more numerous, the closer and closer you expect to see the ratio of heads and tails approach 50%. And the further and further and further you get away from 50%, as you get more and more flips, that becomes more and more statistically improbable. So, the same goes for populations. Imagine we had a large, let me see if I can give you a better image. Okay, I can't find you a good image, but I think you get the idea that if you have a very small population of individuals, and say the allele frequencies, the distribution between the two alleles is 50-50%. If you only have 10 individuals in a population and 7 people die, it wouldn't be weird to be only left with 3 of the same allele, and completely eliminate the second allele from the population. However, if you had 100,000 people and 70% of those people died, it would be very weird to be only left with one allele type, because there's still 30,000 individuals in that population. So genetic drift has a much more pronounced effect on small populations. So therefore, yeah, so I think actually that's it. That's all I'm going to talk about for genetic drift. Okay, so this is also going to be complemented, this little mini podcast, or screencast, is going to be complemented by talking about Hardy-Weinberg equation, which we are going to do tomorrow in class, and we are going to mate in class. It's going to be exciting. So, that's all I have for you today, and I will see you tomorrow.